Não, eu não sei a letra toda. É. Tá, vai. Gonzaguinha, então. Chega. Chega. Lá. Chega de tentar dissimular e disfarçar e esconder o que não dá mais pra ocultar, e eu não posso mais calar, já que o brilho desse olhar foi traidor e entregou o que você tentou conter, o que você não quis desabafar e me cortou. Chega de querer chorar, sofrer, sorrir, se dar e se perder e se achar, e tudo aquilo que é viver. Eu quero mais é me abrir e que essa vida entre assim, como se fosse o sol desvirginando a madrugada. Quero sentir a dor dessa manhã, nascendo, rompendo, rasgando, e então eu chorando, sofrendo, gostando, adorando, feito louca, alucinada e criança. Sentir o seu amor se derramando, não dá mais pra segurar, explode coração. Okay, now we'll do something more boring. So I'm going to finish talking to my lecture on the olfactory system. Okay, so I'm going to put up again the picture and that I drew of the structure of the olfactory system, and then we're going to go and start discussing details of the problem. So so we have a situation where you have, let's say, a bunch of receptors of type 1, a bunch of receptors of type 2, a bunch of receptors of type 3, basically a certain number of receptors of each kind. And then you have these things project to structures called glomeruli in the next stage of olfactory processing. So for example, in mice, this area here would be called the olfactory epithelium, and this would be called the first stage of the olfactory bulb. In the epithelium, what happened? Sorry. In the epithelium, it's not the case that these receptors are sort of organized in little patches like this. They're all sort of randomly scrambled. And there's this interesting developmental, during the retina development of the there's this interesting phenomenon that you have this, the neurons pick a receptor to express rather than having, because they have genes for many receptors. So somehow if you express one, you've got to shut down the others. So there's this very interesting thing about how they do that. Okay, very good. Another interesting thing that will become important for us in this lecture is that these receptors in the receptor neurons, the ORNs, olfactory receptor neurons in the mammalian olfactory system epithelium, have the interesting property that they die every month. They die off, and then they're replaced. There are stem cells that replace it. It's one of the few places, sort of in the adult human, let's say, that you get neurogenesis, right? You get new neurons every month. And we're going to use that as a lever for experimental investigation of this. Okay, so we're going to think about that in a minute. So anyway, these are collective glomeruli. Then there's this very interesting circuit where the glomeruli, they have output neurons called mitral cells. I'll draw them as little triangles.
Wait, I'm having trouble drawing triangles. I'll draw them as squares, lots of squares. So there are lots of these things. They're shared. And so it sort of mixes up the signal. And then there are these things called granule cells. So these are mitral cells. These are granule cells. Granule cells. And I'm going to, and then basically the granule cells take input from the mitral cells and then they sort of feed back and they're inhibitory. So these are inhibitory. Everything else is excitatory. So they provide some kind of feedback inhibition, which is a typical motif you find in neural circuits, especially in sensory systems. So you get this feed-forward signal that's sort of exciting it layer by layer, and then you get this sort of feedback inhibition that reshapes the signal and somehow removes the stuff that you're not interested in. So for example, one thing this kind of feedback inhibition can do is to normalize the signal. So you may not care about the overall loudness, so to speak, of the signal, but somehow just the relative loudness of the response in each receptor. You can implement that as you can use feedback inhibition as a circuit mechanism for doing that. Another thing you might be interested in is, for example, you may not care how bright the light is, but how bright the light is here relative to a nearby region. Or how much of this receptor responds relative to another receptor. And you can, of course, implement that by feedback inhibition. Right? All of these things, this is a circuit motif that does many interesting things. So that's this feedback, there's this thing. Also, they are unfortunately or fortunately connected all to each other. So this is actually a god-awful network of granule cells connected to each other. And so this is an example where sitting down and thinking about it and doing theory, it's actually hard to discern what you think these things are going to do. And this is a case where actual simulation by building a complicated network is helpful, right? So there's a lot of work on that, which we won't discuss because there's not enough time, and that perhaps overlaps a bit more with Professor Copelli's lecture. Then we said that each of these things, then there are random projections to the cortex. And the cortex, there are cells here that are called pyramidal cells, pyramidal cells, pyramidal cells. And overall, there's an excitatory, inhibitory network. And I'm throwing all these kinds of buzzwords at you basically because I don't have time to go through each one of these in detail. If I had 2 weeks, I'd go through this also. And another very, very important thing in the study of cortical circuits is studying these kinds of excitatory inhibitory networks. So these excitatory inhibitory networks, typically in cortex, you'll have 1 or 2 kinds of excitatory cells, and then 5 or more kinds of inhibitory cells, and they'll make very particular circuit motifs. Right? The excitatory cell will have this particular kind of inhibitory cell connecting close to its cell body, another one connecting far from its cell body. Some of these inhibitory cells will take inputs from the previous layer, some won't, some are purely recurrent. Some of the inhibitory cells will take inputs from deeper brain areas, so there's some kind of control circuit. Amazingly, although we know a lot about the circuits, about how they look, very little is understood about how these circuits produce their function. So that's another kind of direction which is very ripe and open for investigation. There may be things that those of you who do neuroscience or theoretical neuroscience will be interested in. Anyhow, this then gets transmitted to some other area that will decide that this odor versus that odor, that this is appetitive, let's say. I'm just giving an example of an odor behavior. Appetitive and aversive, go away or go towards. Of course, that's aversive. Of course, there are many, many more complicated olfactory behaviors than appetitive aversive. I'm just giving an example of a binary decision you might make, go towards, go away. So this is this entire pathway conserved from fly to human to elephant, whatever. And so that is why we think it should be, we should be able to explain it. Now, for lack of time today, I'm going to stick to the receptor layer because that's also closest to the kinds of things I did with CRISPR, and we'll say a few things about it. But both our own lab, as well as many other labs, of course, have lots of work on the theory all the way down, and I think you'll enjoy reading about it. So you can, if you look at my papers on the subject, you can go find them easily, and the reference lists contain references to everybody else's papers, and so that's a useful lever for investigation. Kawe?
Oh, when I was formulating the question, I would ask about the part that you just said that you will not go on. So,
ask the question anyway.
Because when I see the people representing neural networks and the like, I always get a bit confused about how do they know that the processing is actually happening in the in that part of the whole circuitry that they are isolated, because they do not usually talk very much about the output of it and what happens with
the feedback that comes back.
Yes.
Yeah. So that is a perennial problem. It's partly guesswork. You know, all science involves guesswork. You guess something, you try to see if it works and fits, and sometimes you've got to correct it. So for example, an important thing I left out is that pretend that this is all feed forward. Actually, there are extensive projections from the central brain down to the olfactory bulb. Very interesting, partly because the granule cells here are thought to be another site where there's adult neurogenesis. So it's believed, so one idea people talk about is the central brain may control where newly born inhibitory cells go and attach themselves to the circuit. I mean, they help guide that in some way. In this way, reshaping the way the odor representation is here based upon your behavioral experience of odors, right? Maybe some are more important, maybe some are less important. Maybe that's partly guided here. So people have talked about that, right? And so there's some modeling studies and things like this, nothing definitive. But it's definitely the case that you have extensive feedback. My own last paper on olfaction involved thinking about the idea that the feedback from cortex is just random and is just supposed to represent the context of the behavior, that every context of the smell in which you're experiencing the smell is just some random vector. So every context is a random vector. The idea here is high-dimensional random vectors could represent different contexts as just different vectors. And then suppose you need to learn that, oh, in this context, this odor and this odor should be regarded as similar, or in this context, these odors should be regarded as more different. You know, some odors are similar, but sometimes the specific differences matter. Or some odors are pretty different, but actually the differences don't matter. They have the same behavioral valence. So, you know, we have some papers suggesting that the cortical feedback here can provide input of same or different in terms of context, and that can be used via learning and plasticity in this circuit, which I haven't talked about at all, right, to entrain changes in the olfactory representation that bring your mental representation, if you like, of the odor to be more similar or more different. And we make some predictions that reproduce things that the people have seen in experiments, both in behavior and in recordings in the cortex. So this is a paper, that's a paper, that's a paper by my student David Curson, my former postdoc, Gaia Tavoni, who's now an assistant professor at Washington University, St. Louis, and myself. You'll find it in PLOS Bio. And you'll also find lots of references to the, I remember my own papers best, but of course there's lots of people who are doing very good work, and you should go look at the references in these papers.
So it deals with the representation of the inputs
yeah, so this is, the receptors are responding.
Okay.
They cause this thing to respond. They cause the mitral cells to respond. The granule cells reshape the response of the mitral cells by sort of inhibiting this and that. So there's a reshaped representation of the input. That representation goes to cortex. Something happens. It's reshaped by the circuitry. And then you have a reshaped representation of the odor world, right? This is your mental representation of smells. Then that thing is processed by deeper layers of the brain to allow you to make decisions, you know, do this, do that, and remember things, and so on and so forth. Super cool thing to think about. So that's why I started talking about how it's well known that your expectations and the context in which you're sensing something can cause you to feel, to perceive something in a different way. The same smell in two contexts may sort of feel different to you, be perceived differently. We know this all the time. Expectations can shape things. So if you expect to smell something, you'll smell it, or you'll tend to, right? If there's, you can fill in missing bits of evidence for something being there, stuff like this, right? So how does this even happen? Very interesting. So some of this has to involve feedback from deeper layers of the brain that form those expectations, are producing representations of the context in which you're behaving, the outcomes, you do something, did you get a good outcome or a bad outcome, right? That has to get fed back to the early circuits. And in the olfactory system, a lot of these things get fed back all the way to the second synapse here. So that's unlike most, for example, vision, where that kind of stuff, that kind of plasticity and that kind of feedback mostly happens inside the brain, inside the central brain, right, in a cortex. But in olfaction, you get these feedbacks all the way back to the second synapse here. And which is why I'm interested in the system, because some of these things that seem very, cognitive or deep or something, your context influences what you perceive, are actually already happening at the synapse, presumably because of the cortical feedback. But almost nothing has been done to elucidate this. So it's an open field, right? There's a little bit of work here and there. And so definitely, if you care about that kind of thing, you should look up the existing work. And there will be, if you use your imagination, in an hour, you'll come up with 10 things that are interesting to do. You're basically here limited by the amount of time you have and your imagination. This is a good system to play in. Go on. Okay, but I'm going to mostly work on the receptor level just because of the amount of time I have and continuity with what we said before. Also, remember, given the vastness of biophysics as a field, my goal has been to do different kinds of analyses that involve different kinds of techniques. So I'm going to do 2 kinds of analyses that involve 2 other kinds of techniques. Today, one of them being information theory, and one of them being molecular biophysics. Molecular biophysics is a very old-fashioned topic in biophysics. And so people it's boring. But actually, it's extraordinarily useful, and I want to demonstrate that. So you should know the basics, is what I'm trying to say. The neural circuit stuff, there's other lecturers who are going to come who are going to give you some ideas about neural circuits. You'll be able to, and then you can read about olfaction, learn what they say about neural circuits, and do what they do in the olfactory system, and you'll be the first to do it. Okay, so here we go. So I'm going to start off by first of all talking about receptor. It's all good? Okay. Okay, I'm going to talk about receptor response models. So as you'll see, I'm sort of mimicking the kind of logic and pattern that we use to discuss CRISPR. And here is a simple linear model. This is the model with which we'll do calculations in a bit. But first, I'll write it down. And I might say that the receptor type A, responds to a odor concentration vector C. Remember, we had said Ci equals the concentration of ith odor. Let's say, and we're going to say R vector equals R1 through Rm, where M equals the number of receptor types, and Ra equals the summed response of Ka neurons of type A. So basically, I imagine a neuron expresses the receptor type A, so I'm going to call it type A. And let's suppose there are Ka neurons that produce that. And remember how this signal gets summed here, pretty much. So I'm going to take the aggregate response as a summed response of all these receptors glomerulus and call that Ra. All right? So we're going to take that summed response, and we also have some constraint that says sum on A of Ka is equal to K, and K is the total number of receptors. So let's set up a model of this kind. Okay. And then I'm going to say K. So your naive linear model here, if you're going to write a naive linear model, would be to say, well, I'm going to sum SAICIKA. So S is going to be a sensing matrix. And if you like, the ith entry of the sensing matrix tells you what you're expecting is the firing rate of the 8th neural type in response to the ith odorant. And then if you double the concentration, you get double the output, right? So this is at a first cut. It's not going to be a stupid model. It won't be great, but I'll tell you what. Well, it'll be a fine in some regions. I'll tell you later in a minute. And then because I'm summing it, I get a Ka, right? Because I have Ka such neurons. And I'm going to add to this, neurons are noisy. right? There's sort of processes that are kind of stochastic in them. So I'm going to add to it a thing here that I'll call, this is Gaussian noise. Gaussian noise with variance, let's say, sigma a squared, because I'm going to leave open that for whatever reason, different receptors may have different noise levels, right? They may be better or worse at producing the signal. And then I'm going to multiply this by square root of Ka. And I'm multiplying by square root of Ka because I'm assuming that this is independent noise in each of the neurons whose responses I'm summing. So of course, remember, if you sum up independent noise, then the amplification of the noise that you get goes like the square root. So this goes like the square root, and this goes linearly in Ka. So you're getting a little bit of benefit by the summing to beat down the noise. Okay? So this could be your baseline model of what these neural responses are. So having written this baseline model, which is what we're actually going to use, I first want to tell you in which ways it's wrong. And I'm going to build a much better model of the responses and nevertheless use these for calculations by linearizing the better model in some regime. So in fact, responses are nonlinear. And you know that because you can't possibly have a neuron which has a linear response to concentrations in an arbitrary range. That just doesn't happen because neurons, if nothing else, will saturate the response. So for a single odorant, you already know that if I take a receptor response function, a dose response curve, if you like, here's the response and here's the concentration, you're going to get curves that are like this. There will be some threshold below which you will tend for basically any kind of receptor. It's going to be kind of flat for a bit, and then it's going to rise, and it's kind of linear there. And then there's going to be a saturation level that happens there. So this is pretty much true for any receptor you choose to name. So at best, a linear model is going to kind of work here. Right? Okay, that's for starters. But secondly, actually, there are more effects. Whoops. When you talk about mixtures, there are more kinds of nonlinearities that you could imagine. So let me draw some kinds of nonlinearities that have been observed. And these nonlinearities, by the way, have to some degree been observed in receptors. And they've certainly been observed perceptually, right? When you mix odorants and you get people to sort of report about them, you find these kinds of effects. So one class of effects is what's called overshadowing. And overshadowing, it goes like this. So I need to, okay, so here we go. So suppose I have odor 1. So, okay, how to do this? I need 3 colors, and I don't have 3 colors. Okay, let's do this like this. So we're going to do, this is odor 1. And let's suppose this is odor 2. And then you take odor 1 plus 2, you put them together, and you get odor 1 plus odor 2. So this is the response curve. So it turns out, although you have 2 odorants, basically if you put both of them in the same time, it'll respond as if it's responding to odor 1. So that's called, so you're overshadowed by odor 1. You can get the reverse effect also. Suppose you have odor 2, and this, let's say, is odor 1. You can get odor 1 plus odor 2 being like this. So this is both called overshadowing. So that's one effect. Another kind of effect you can get is called suppression. And suppression goes like this. So you have odor 1, which goes like this, let's say. Then you have odor 2, which goes like this. And then you present them both at the relevant concentrations, and you get something that goes like this. RC. So this is a situation where it's not that odor 1's response kind of dominates the whole thing, but rather somehow the presence of odor 2, well, although odor 2 by itself produces a response, you put them together, odor 2 sort of pulls down the response that you would have had to odor 1. Sort of like an inhibitory thing.
Yeah, Professor, in these graphs, the concentration of O1 and O2 was always the same?
Yeah, so you take this and you keep it going like that.
Okay, so the line O1 plus O2, they have the same concentration?
Oh, that's a good question. You're asking whether or not O1 plus O2 is given at the concentration specified here. So what I'm trying to do here is I'm trying to take the concentrations as seen on the x-axis for both O1 and O2 and just give them both.
Okay.
Right? You could do a different thing that has more molecules than O1 or O2 separately. So you could sort of scale it down, right? You could sort of reduce the concentration of both. So the net concentration of molecules was the same. That's not what I'm doing here,
Okay.
which is why the naive additive expectation is that you add them and you get a much bigger curve. And I'm sort of illustrating situations where that doesn't happen.
Okay, thank you.
Yeah. So then the third kind of thing you can get, there are many, many such effects, but I'm going to illustrate a few. There's synergy, and synergy goes like this. So you have, let's say this is odor 1, let's say this is odor 2, and then you put them together and you get this. So O1 plus O2 bigger than, well, the response R of O1 plus R of O2. So this is a situation where you had the responses of each one separately, and you put them in together, and then you get more than the sum, right? Which seems really particularly peculiar, because where are all the molecules coming from? Yep. Then there's another effect called inhibition. Inhibition, which looks like this. So you have, let's say, this is O1, and let's suppose that this is O2. And you put them both together, and the response is less than both. So, right, this was a case where the response is bigger than the sum, and this is a case where the response is less than the response to either of the odorants. So this seems like a total zoom of effects, which certainly are not captured by the linear model. And what I'm going to try to argue to you is actually, these kinds of things are very well known in old-fashioned molecular biophysics. We can very easily make the absolutely simplest model in molecular biophysics, which will actually reproduce many of these effects. Some possible effects, which I will mention, are not reproduced by that minimal model. And if you see them, you have to add more bells and whistles to the detailed receptor model. So just to illustrate, we're going to do that little calculation. Okay, so here's the calculation. So how can we capture such disparate phenomena? Okay. So this is described in a paper by Vijay Singh, Nicole Murphy, Joel Mainland, and me. I think this is in PNAS, and I think it's 2020 or so. Okay. So basically, what we would like to do is we realize that if you want to make a faithful model, I mean, this is all setting up for a future, excellent model of everything in olfaction that isn't linearized and things like that. To do that, you do need some kind of thing that mimics these nonlinear responses. Part of our problem is that if you look at these things, it can be that odor 1 synergizes with odor 2, but is inhibited by odor 3, and then odor 2 and odor 3 have some other weird interaction. It just sort of goes on like this. So what is an experimentalist to do? Right? Suppose you've got 50 odors you want to play with, and 100 receptors. You have to try all possible combinations of these things, it feels like, to figure out what's going on. That's just not going to happen, right, in the lifetime of this universe. So given the numbers involved. So you need a method you need a method. So one of the roles of theorists is, when faced with problems like that, to propose methods where you can do minimal measurements of some kind and then use those minimal measurements in a good model of the system to get all the results you want. Okay? So that's our goal here. So basically, ideally, we want to measure dose response curves. And ideally, we should not even have to do curves. So this is dose response curves for single odorants. And derive the mixture model. So even more ideally, we would know enough about molecular biophysics and the shapes of these molecules that we would say, aha, you tell me which molecules you're talking about and the receptors you're talking about, and my model will tell you what the response is going to be, because then you don't even have to do those measurements, because even dose response measurements for many receptors and many odors take a very long time. You have robots doing them and things like this. It still takes a very long time. And it's possible that these sort of somewhat magical machine learning techniques that have been come to light in doing things like protein folding and stuff like that could be deployed for this kind of thing. You somehow figure out, if you have this shape and this shape, this is going to be the response curve, right? So that might yet come, and if so, great. But while we're waiting for that to come, let's assume that an experimentalist has given us these dose response curves, and I'm going to suggest a way in which you can make a simple model of the mixture response just knowing that data. Okay, so the simplest scenario is for receptor binding and activity of this kind is competitive binding. That is the absolute simplest thing that can happen. This means you have a receptor, that's the binding pocket. And let's suppose there are two kinds of odors to start with, odor 1 and odor 2. And each of them can bind or unbind, and I'm going to call the rate of binding K1, and R1 is the rate of unbinding. And O2 can bind or unbind. So we have R2, K2. And you can produce a bound receptor in this way. I'm going to start writing down some equations underneath for what these processes mean. Actually, let me write that here first. I'll say O1 plus the receptors go to K, go this way with K and this way with R1 to make a bound receptor OR. So that's a complex. And O2 plus R has binding rates K1, K2, and R2 to produce O2R. Okay, that's the first step. Now, in practice, what actually happens is the bound receptor by itself, once you bind it, doesn't actually produce a response. A further changes to typically happen. And so you have here this bound, so this is the O1R, this is the O1R complex. And there's a second rate, K1 star this way and R1 star this way, to put this into what we'll call an activated state. So this is the activated state. And when the receptor is in its activated state, it starts to produce responses. So there's a two-step process in these things. So there's similarly for O2R. So let's write that process down here. So I should draw a second pair of arrows. There's K1 star and R1 star. And this produces O1R star. This is the activated form of the bound receptor. I have K2 star and I have R2 star. And I'm going to have O2R star. Okay. So what are we supposed to do to model this? Well, we write down the rate equations for these chemical processes. I mean, they're very much like the population models that we were doing before, but these are just chemical rate equations. So let's write them down. So what is the rate of change of the density of receptors? dr dt will have one term, which looks like minus, because this is reducing the number of unbound receptors, right? dr dt is going to be equal to minus K1 C1R plus K2 C2R. These are the two processes that bind receptors. But of course, receptors can also unbind. So you have R1 plus R2, R2. Okay? So by this notation, R equals, I'll put this here, unbound receptors, and this is the concentration of O1R concentration, and this is the concentration of O2R. Okay, so that's the first equation. Then we also note that the receptors which have been bound can proceed to activate. So let's write down the rate equation, the chemical rate equations for the activation. So first of all, I can increase the number of receptors, of bound receptor complexes as K1C1R, KI-CIR for the ith receptor type, right? Because it's type 1 or type 2. And you can also increase the number of bound complexes by deactivating an activated receptor. Okay, so this is like, you remember with the CRISPR, there was the wild type, there was the immune wild type, there was the infected immune wild type, you know, there are these sort of various cascades of these things, and they're just rate equations going back and forth, same kind of thing. So I put it down here, and then I get to lose these things either because they become activated, that's this term, because they become activated, which is this term, the second one, or because they unbind the molecule, and so you get that second term there. So do I need to define anything more here? No, everything is defined. Okay, finally, we have dRi star dt, namely the rate of change of the bound activated receptors. There are two ways in which this can change. One is you can make them, so this is the rate for activating a bound receptor, or you can deactivate, you get minus Ri star Ri star. That is the rate of deactivation of the activated receptor. Great. So now we know what we're going to do, right? You can analyze this in many ways, just like in CRISPR and so on and so forth, in this population dynamics. We can analyze, you know, the all kinds of things in these things. Points of stability, rates of change, points of instability, fixed points, you know, just a dynamical system of the usual variety. And there's an enormous literature on how to analyze this. For the purpose of this lecture, and because of the shortness of time, we will just do one thing. We will look at the steady state, right? That's always easy to do. So we'll look at the steady state, because that will tell us how active the receptor is supposed to be in steady state. So here's the steady state. So the steady state is given by solving for all three of those time derivatives star equals 0, and that reduces these algebraic differential equations into a bunch of algebraic equations. And as you can imagine, at that point, it's just algebra to rewrite these things in some form that you find convenient. So that's pretty tedious, so I'm not going to do it on the blackboard. Also, there are n different books you can find which will just do all the steps out for you. And this is what you get. So we're going to agree that the response, which I'm going to name F, is going to be proportional to the total number of bound activated receptors. So this is going to be R star equals R1 star plus R2 star. You know, someone asked me to hand out my notes, and I'm going to ask them to scan it. But I just want to point out that there are typos in my notes, so you'll have to use your judgment and compare against your notes. It's mostly typo-free, but I just noticed that I don't have the star in my notes, so there'll be some typos like that. Okay, so, well, fine. So I'm going to write down an equation for what this F is going to be. So F is going to be proportional to R1 star plus R2 star in steady state. So it's a matter of doing the algebra to solve for that steady state, plugging it in, and this is what you get. So you get F is going to wind up being, the response is of course a function of the two concentrations. And the equation you get as a function of the two concentrations is written this way. So there's an F max, which is proportional to the maximum number of receptors you have, which is some number. And then there are going to be two terms. One is the concentration of the first odorant divided by what I'll call EC51. I'll tell you what that is in a minute. Plus the concentration of odorant 2 divided by EC52. And EC50 is equal to the concentration at which the response, at which the single odorant response is at half max. Okay, so that's something you can measure clearly from the data. Times E1 here and E2 here. So E1, EI is what I'll call receptor efficiency. So it tells you the conversion factor between having a concentration and a response. Clearly this has something to do with the binding affinity. And I'm going to define these two in a moment in terms of these parameters K1, R1, etc. so that we have it for reference in terms of the fundamental rate equations. But these are just numbers you can just measure, right, from the dose-response curve for a single neuron. That's why we're parameterizing this way. You can measure it without knowing the chemical parameters underneath. But I'll write it in terms of the chemical parameters too. And then you get to divide this thing by 1 plus C1 over EC51 plus C2 over EC52. Although we can measure the efficiencies and the EC50s directly from the data, I'm just going to write out what the EC50 is. So EC50I is equal to RI star over Ki divided by Ki star plus Ri star. So in the algebra, it's this combination of parameters that I'm naming EC50 because this combination tells you the response at half max. Concentration for half max response rather. EI likewise in terms of the microscopic parameters is Ki star over Ki star plus Ri star. So as promised, this has something to do with comparing your binding rates versus your unbinding rates. So it has something to do with how well the thing is going to sort of capture the molecules, how efficient the thing is at grabbing and keeping the molecule. Okay? So those are these two parameters. We understand them microscopically. Your experimental friend can measure them, and therefore you're good, right? I mean, you can use this to propose a model of the binary response. Real odors, as we said in the last lecture, can have as many as 40, 50 different odorants in them. And so you can redo this analysis if you wish for larger mixtures. And the answer you're going to get is obvious because clearly you just sort of add it up. So you're going to wind up with the answer that F for a collection of concentrations, and this is what we care about, is going to be equal to F max times a sum on all deodorants, I equals 1 through n, of EI CI over EC50I over 1 plus sum on I equals 1 through n of CI over EC50I. So to me, the most amazing thing in working on this paper in some ways was that nobody had written this down before for the people who do olfaction. Right? It's not like this isn't known. This is completely standard competitive binding. But no one had applied it to olfaction in the year 2020. Right? That's pretty far along. And everybody was using a simple linear model of odor response summation, which I'm going to use too. But I was all saying, well, it's going to be very difficult to measure all these parameters. Well, if you have, this is where physics is useful. If you have an actual model of what's going on underneath, you can really, save a lot of time. You can make things much simpler. So, okay, so that's our description of the nonlinear response function. So the question is, how good is it? Because there are many other phenomena that could happen. I'm going to list 2, but I'm not going to describe, and we can model all of them and include them, but I'm not going to describe that for lack of time. So there are, sorry, let me write it as we could include other effects. Let me give you examples of 2, and then we'll move on. The other effects we could include are, for example, we could say that we want to include facilitation. And facilitation is a process where binding of O1 somehow helps binding of O2. Maybe there are 2 different binding pockets, and sort of binding one opens the other, or maybe it's that there's one binding pocket and has room to fit 2 things, kind of. Who knows exactly, right? But this is a phenomenon that you could model, and which can happen. You could have multiple, as I just mentioned, you could have multiple independent binding sites. So what if it's the case that O1 and O2 don't actually fit in the same binding pocket, but they're separate, and you can fit them both in. And so it's just, you just have to stretch your imagination for what kinds of phenomena you might expect. And indeed, people have been thinking about this a long time. So there's long catalogs of the kinds of effects you might expect, and we should expect all of them to happen, right, at some level. Because of that, the real question is, how well does this work? It could be that even with all this effort, it's kind of useless, because most of the interesting stuff is facilitation, right? So let's check. So one of the challenges with checking a thing like this is if you go to your experimentalist friends and say, please give me data on dose response curves for for as many receptors as you can, as many odors as you can, and for all the mixtures in different proportions, they'll go because even if they're willing to share, which they're not always willing, the dose response curves are easy to do. But just think, if you have two odorants, and you want to measure the responses of this neuron to 10 different proportions of each one, right? You're already talking 100 combinations, right? It's just a lot of work. And for things with olfaction, one of the problems with doing experiments is you've got to feed the odorant, right? And so odorants are actually all slightly sticky. They stick to stuff. So you have your tube, you pump your odorant through, now it's got contaminated by that odorant. Now you got to clear the stupid thing out, right? I mean, this takes forever, right? So because of this, these are hard experiments to do, and you really need to plan everything in such a manner that you can convince them that this is going to work. You can get useful data from just a little bit of things. And so I'm going to show you some convincing results. So the first thing you can do is, before you can try to convince your experimentalist friend to do an experiment with large mixtures with 12 components and things like this, for them 12 is already very large, you want to get them to do binary and ternary mixtures, which they think that they can handle slightly more easily. So let's say that there are 3 of these dose response curves. Let's say for 3 molecules, and this is the case from our papers, there are 3 molecules known as eugenol, acetophenone, phenone, and coumarin. So they measured these kinds of dose response curve, coumarin, and they measured dose response curves for these 3 molecules. So you would think that if you do this, well, you just have to read out these parameters. But as it turns out, because of the experimental conditions, it was hard for them to measure the saturation point. So then you have to go into lots of model fitting and error bars, and do you believe you're fit? You know that kind of thing. So you do that. Let me just finish with one more thing. So then, if you have this data, well, you do your fitting very carefully, and you pull out these 2 parameters. In principle, you can predict the responses using the competitive binding model for any pair or triple of these things, into any proportion you want. And it turns out that for 12 receptors that we had data for from these folks, there is a very excellent match, real response, and this is the CB prediction. For 2 or 3 receptors, it wasn't such an excellent match. And there were signs that there were things like facilitation, inhibition, and things going on. But now we have a technique for trying to figure out for which receptors you need to do that modeling. You fit it with something much better than the linear model, with the competitive binding model. And if it doesn't work, if it's above or below, you know what sort of effect to look for, and you can go look for it. I'll say one more thing, and then I'll take your question. So then the third thing you can do to really convince people, you need to do something where you look at you know, many odorants at the same time, because really the goal here is to try to push the neuroscience of olfaction into regimes that resemble real-world olfaction, which involves 40 component odors, not one odorant, etc. That's why we're doing this. So this is dose-response curves for 12 odorants for one mouse receptor, OLFR, let's say 168. Doesn't really matter which one it is. So, but I'm trying to illustrate, part of the art of being a theorist is really milking everything you can from the limited data that's sometimes available. So, this was really difficult to convince our experimental collaborator, who's an expert in this kind of thing, to make these measurements of 12 dose-response curves and like this. But then now you can study complex mixtures. And once again, I wouldn't be telling you this if not for the fact that there's an excellent match, a very good match anyway, of so this is the competitive binding model, the measured response. So there's a good match here, and the root mean square error is equal to the root mean squared error of the linear model for this case divided by 10. So there's a tenfold improvement in this sense compared to what a simple linear model would say, and that includes all the details of these complicated interactions between these molecules. So this is an example of how traditional old-fashioned techniques like molecular biophysics are actually really useful. That's why they're invented. You should know them, right? And when necessary, you should use them. Or if you don't know them, know that they exist, so that when you need them, you can just go learn it and do it. So that's just my homily about that. And so that's a different kind of biophysics technique that you've just flashed. Question?
Just to be sure, with this very simple model, we can have all these effects like overshadowing,
yes.
inhibition.
So this, absolutely. So this very simple model by itself, but for different values of the EC50s and efficiencies, can show overshadowing, it can show suppression, it can show inhibition. I don't remember what other things I talked about. Oh, synergy, it can definitely show synergy. So which is kind of surprising because it's very, very simple. But in fact, indeed, if you just play with these parameters, just explore the parameter space, all of these effects are there already in it. That doesn't mean the particular instance of synergy you see came about because of this. But that should be your default guess. This is a better default guess than better null model, if you like, than the linear model, is what I'm trying to say. That's just, the linear model was fine for the 20th century. This is a better model for the 21st century. And in 50 years, there'll be an even better null model with many more details in it. Okay, all right. Okay, very good. So that was that. I'm going to erase this now. So with this said, I want to try to address a question in the olfactory system that resembles the kind of question I described for you in the CRISPR system. That's partly to discuss the point that similar kinds of questions appear repeatedly in different kinds of biological systems. Like, for example, you could ask, when you're asking questions about diversity in a system, well, if you have an ecosystem, you could ask how diverse or not diverse is it, and what are the parameters that are determining that. In CRISPR, we could have talked about it as a sort of ecological kind of problem, and how diverse is that. And right now, I'm going to talk about how diverse should the olfactory repertoire be. Right? So I'm going to take that class of questions. So, let me change pages. I have like half an hour. Perfect. So we're going to ask questions about the organization and diversity of the receptor repertoire. which you'll recognize sounds very similar to the word just said about we'll ask questions about the organization and diversity of the CRISPR spacer or immune repertoire, right? So I'm going to ask a similar kind of question here. So in the case of CRISPR, we basically were trying to argue that the repertoire ought to be adapted to some aspects of the pathogen landscape, and we use very simple things about the pathogen landscape, and to the constraints of the system. In that case, for example, how many Cas proteins you had, avoiding autoimmunity, and things of this kind. So here, we're going to ask, suppose you have, well, basically, first I'll start writing out the details, but really the idea here is going to be, instead of the pathogen environment, there's an odor environment. Suppose there's an odor environment, and your goal is to have a sensory system that senses that particular odor environment well. How should you organize the distribution of receptors to, with respect to the odor environment? So that's very similar to the CRISPR question, but you'll see we'll use a different class of techniques from information theory. I'm going to use this as a means of introducing a little bit of information theory as another method. Okay? So we're going to say, suppose there are n odorants. Remember, n, we said in the last class, is something of order 10 to the 4th perhaps, or maybe 10 to the 5th. And C equals C1 through Cn is the concentration vector. So the idea is, we're going to think about this as saying that the environment is going to be defined by having some distribution over these things. Obviously, by even saying that, I'm making some kind of approximation, because as I've already said, even if I have a certain odor in the game, as I move my nose around, there are odor plumes, it's all very dynamic, stuff like this. I'm leaving all of that out. We're going to sort of average a little bit over time. And imagine there are smell episodes. It's not entirely true because you're moving through the world continuously, but I smell, I don't know, the jackfruit. Then I smell, I don't know, the petroleum fumes. We're going to imagine this as a sequence of discrete episodes, which is not entirely true. But anyway, that's a step up from where we are, which is right now we can't model anything. So we'll step up, and then we can move to the more continuous thing as time passes. So we're going to let m equals the number of receptor types. Same notation that we were using earlier. So we have R vector equals R1 to Rm equals the responses of the types. Remember, we're going to take the summed responses, where we sum over how many receptors you have of each type. And let me remind you again, K equals the total number of receptors, and we have Ka is equal to the number of ORNs, olfactory receptor neurons, of each type of type A. Okay. So here, of course, the response is some function of the concentration vector. So we need a little model of what that thing should be. Okay? And I'll describe that in a minute. But before I put out my model, because the model I'm going to make approximations, right? I'm going to make it linearized and everything. I'm going to first phrase the problem I want to solve in a way that's not dependent on that specific model. But then we'll incarnate the thing we want to do in a specific model. So the question set up this way that we want to ask is, how should the distribution of receptors, which we can think of as being a set K1, K2 to Km, be organized to best represent the odor environment. Okay. Which, of course, raises the question, what does best mean? And that's what I'm now going to try to formulate. Okay. Okay. Time, good. All right. So, okay, so here's an idea. Let's use the amount of information, and I mean the word information in the same sense as the phone or internet company, bits. That the responses give us about C. So the idea here is that the responses, give you a certain amount of bits of information. The deeper part of the brain does whatever it has to do with this. But if there's no information, it can't do anything. You have lots of information. The brain is a pretty big thing. Maybe you can use it well in some way. We could ask more refined questions about how that information should be formatted so that the computational device further in can use it well and things like that. Those are also very nice questions to ask. But let's just start with basics, right? Is the information there? So we're going to use the language of information theory. So in information theory, you imagine that there is an incoming signal. For us, that's the odor vector. Then you say to yourself that there is an information channel. For us, that's the kind of nose, right? The nose, the olfactory epithelium, and it gets into the olfactory bulb. That's the channel through which the information is passing. And then you produce a response R. And the response for us is, the responses of all of these receptor types. So first, let's suppose that R equals S. So this would mean that for us, there's somehow as many receptors as there are odorants. Each receptor is completely specific for every odorant, right? That kind of thing. Each receptor is completely specific for every odorant, and it just passes the information through. Right? So you're basically directly looking at an image of the odor environment. You're not. We'll come back to that in a second. But suppose this was the case, what would you do? So in particular, so S, the signal, is going to equal, let's say, X1, X2, X3. There's a series of sort of episodes of this kind. And the episodes, let's say this is at X, at T equals 1. Actually, I'm going to use a different notation here. X at time 1, X at time 2, X at time 3. Maybe I should even call it C. I'm keeping on changing notations, right, etc. And that is the odors that I'm going to measure, I'm going to see at each moment. It's actually more than a vector. It's an infinite sequence, right? So information theory is often formulated in the language of an infinite sequence of things you're going to send. It's like as if you're talking forever. And then what you do is you measure the information rate per second, how much comes through the pipe. That's like the internet company saying, we will give you 5 megabits per second, right? That's the number that they're computing, how much is getting pumped through. Okay? So, and let's also suppose each is an independent draw, each C is and I've removed the vector sign here, by the way, for the moment, is an independent draw from P of C. We don't have to make them independent. You can have correlations in here, and then there's a thing you have to do. There's all kinds of techniques in information theory for thinking about this when there are correlations. But let's not, because this is not really an information theory class. There's a wonderful book by Cover and Thomas where you can learn all of these things. Let's just take these to be independent. That's like the jackfruit smell and the petroleum smell were independent of each other. You just happen to walk past one and the other, that kind of thing. Right? So let's take that assumption. So if you agree with this, then the number of bits per time of data in S is quantified by an object and quantified by an object called the entropy. And I'll comment on the relationship with the entropy in physics and thermodynamics in a minute. But for the moment, it's just a word, right? The entropy, which is defined like this. The entropy is minus the sum over all the things that you could send. So it's the probability of C sub I. Remember, C sub I is the ith order. Now we don't have order vectors for the moment, right? I'm just, for simplicity, I've removed the order vectors. It's just an orderant. Log P of Ci. And this can be any base, but base 2 gives you bits. Right? The thing that you call bits is when you take base 2. So, of course, in some situations there are continuous things. For example, you may imagine C, the order, this variable is somehow continuous as opposed to discrete, in which case you would write this, the integral dc P of C log P of C. So I'd like to make a couple of comments about this. One is, this entropy, for the physicists in the audience, is the same entropy that we have in statistical physics. So, for example, if you apply this formula for the Boltzmann distribution, on the Boltzmann distribution in the canonical ensemble, for those people who have studied that, you will get precisely the numbers that you, the formula that you will get for the entropy in other ways, in thermodynamic ways. It's the same object. However, the meaning here is different. So in entropy, in statistical physics, sort of entropy is some kind of disorder. It has to do with microscopic motions of things that you can't access. But in information theory, suppose you have a message of length L. So you have this channel, and you wait there, and it sends you C1, C2, C3, and you get L such messages. You get L of these things, or you smell L odors. Then the statement is that E to the LH equals, for large L, so L tending to infinity if you like, for large L, E to the LH is the number of distinct messages that can be sent. We're going to check that very quickly and qualitatively on this board, and then I'm going to move on. Suppose, for example, right, with probability 1, you have order 1, and no other order has any probability. You always smell jackfruit. Then what's going to happen? Well, the first one is jackfruit, second one is jackfruit, third one is jackfruit, and goes on like that. Clearly, you send exactly one message. So if you look at this, the entropy will give you 1 log 1, which is 0, and 0 log 0, which is also 0. So the entropy is 0. So E to the 0 is 1. So you get one message. Suppose you can smell jackfruit or petroleum, and you're equally likely to smell both. So this completely random sequence of jackfruit and petroleum. Then each of them is probability 1/2. So you'll get 1/2 log 1/2 plus 1/2 log 1/2. If you check that out, that's log 2. So if you check out log 2, well, if you stick it in here, E to the L log 2, this will turn out to be 2 to the L. And of course, we're completely randomly sending messages. There are 2 to the L possible messages. So those are two limits you've just checked. And that is the meaning of entropy here. And you can discuss why that meaning of entropy actually connects well with the meanings in statistical physics, but that's not our job today. Okay? So good. So this object, the entropy, in some nice way quantifies how much it's telling you, right? If R equals S, and then you take this entropy, which will be the entropy of S, you're just figuring out how much, how many different things it could tell you. That's a nice notion of information, how many things you could learn from it. But of course, in real life, it isn't that simple. So let's add the complications of real receptors and real information channels. And I'm going to illustrate ways in which the response need not be the input. Okay. So it can be that the output does not faithfully reproduce the input. So let me illustrate at least two ways in which this can happen. Imagine that the input can take 4 values and the output is only able to take 2 values. This is actually standard in sensory neuroscience. So for example, the light levels can vary by I think 9 orders of magnitude or more between midday and the dusk or something like this. Actually even more than that I think. But your neurons only have 1 order of magnitude of dynamic range. It's like between 1 and 10 hertz, right? So there's a massive, massive compression going on. So this is a sort of compressive effect. And in this compression, for example, here's an example. Both S1 and S2 are encoded or transmitted as R1. And both S3 and S4 are encoded as R2. This is basically a sensory system, maybe an olfactory sensory neuron, that can report to low versus high concentrations in the 2 responses, but can't give you any more detail. Right? That could happen. Right? So that is one way in which you can lose information going from input to output. You just don't have the dynamic range in the output to represent the input. Another way you can lose, so this is the input and this is the output. Another way you can lose information to the input and the output of a channel is as follows. Suppose you have 4 inputs, and I actually this time have 4 outputs, so it was perfectly possible and by the way, if you're talking about neurons here, you may talk about R1 through R4, maybe response level 1, 2, 3, 4. How often they're firing or the magnitude of their firing rates and things like this. Okay? So now suppose you have a situation where sometimes the signal goes straight through and sometimes it changes due to noise. So here you have noise. So for example, here the probability of getting R1, given that the input is S2, is let's say epsilon. The probability of getting R3, given that the input is S2, is epsilon. And the probability of getting R2, given that the input is S2, namely the faithful transmission, is going to be equal to 1 minus 2 epsilon. Right? So this definitely happens in sensory systems. There's noise all the time. You get the wrong response, if you like. And we can describe this, for example, using, in this case, using a noise matrix, just to visualize this. So I would put down here a 1 minus epsilon for the input 1, and epsilon 1 minus 2 epsilon for the input 2, epsilon 1 minus 2 epsilon for input 3, and epsilon 1 minus epsilon for input 4. Right? So that sort of represents the noise, and each of these tells you the probability that a given input leads to a given output. Okay, so now what do? How to quantify information. So we are going to use a very important quantity, which has many properties which we don't have time to explore. So what I'm going to do is I'm going to introduce the quantity, then you know it's there, and you can check out more properties in n different places, including Wikipedia. But again, Cover and Thomas's book is excellent for this. So you have a signal, comes into a channel, goes out as a response. We agree that the response may be noisy, may be compressive, or otherwise lose information. But what we have in our hands is the joint distribution, let's say, of S and R. Joint distribution. And of course, from this joint distribution, you can extract many other things. For example, if you like, the integral dr p of s comma r, and this would be a sum over values of r if this was a discrete distribution, is the marginal distribution p of s, right? That's by definition of joint distributions. Likewise, you can extract from this p of r. And you can also do things like Bayes' rule. So Bayes' rule tells you that the joint distribution is the conditional distribution p of s given r times p of r. Or you can factorize in the other way and write it's p of r given s given p of s. So with these objects, you can do all kinds of interesting manipulations, some of which we will do. So what we have is the joint distribution now of the input and the output. So we measure, so we quantify, how to put this. I'm going to define a quantity called the mutual information. And the mutual information is supposed to tell you how much, actually, I'll write that down in a second, okay? So I'm going to write down I of S comma R, which is going to quantify how much S tells you about R. The input tells you about the output. And this turns out to be the entropy of R. So you compute the probability of the responses and you compute its entropy, plus the entropy of the input. So this is the output entropy. And remember, once you have the joint distribution, you can work out the distribution of R and compute this in the way I previously described. This is the input entropy. And then you subtract from it the joint entropy. So this is the entropy of the joint distribution, which we said you have. If you want, you can use Bayes' rule and these factorizations and things and write this in many different ways that may be more convenient when you're actually calculating. But this is the basic definition you can work with. And you can see it's symmetric. So this is also equal to I of R comma S, which is the how much R, the response, tells us about the input S. And that, of course, seems like a good measure of how good the channel is, right? How much did you learn, right? And as you know, if it's going to tell us this in bits, following the analogy with the entropy that we were describing earlier, roughly speaking, this is going to tell you, if I read the response, how many different inputs can I discriminate? So I'm an animal, often I have a discrimination task. I want to tell the inputs apart from each other. That's why I have a sensory system. Well, how many different inputs can I discriminate given the output? And this quantity answers that question. So it seems like a good object with which to reason about this system. Okay. So great. So what I need to do is I need to make up a little model for the olfactory system in which I measure the mutual information between input and output. Ideally, the experimentalists have measured everything, for every possible input. Well, ideally, they've told me what the odor environment is because they've measured it. Then they've told me for all the molecules in the odor environment, here are the responses, and they've measured it. So I have the joint distribution in my hand, right? And if I had the joint distribution in my hand, which, in some future date, you could imagine getting a hold of. You use sort of molecular spectrometers and stuff to measure the molecules in the environment, see what they're like. You measure the responses somehow by embedding something inside the nose. You could imagine all kinds of things, right? So this is possible. So one day we could get that distribution. We don't have it, so we're going to approximate our way through this. So let's simplify our model, our olfactory model, so that we can just do a calculation to get a sense of what qualitative effects we're expecting to see. Remember, as I've been saying, you know, sometimes when you don't have enough data, you have to have the art of describing things that are sort of true. And we're going to find a way of describing things that are sort of true. It's mostly pictures after a moment, so I will get through this, I think. So the first thing I'm going to assume is the probability distribution. So this is the environmental probability distribution. Is a Gaussian. You knew I was going to do that. With some covariance matrix. This isn't all bad, because it's probably the case that jackfruit is a little Gaussian, and that petroleum is another Gaussian. And so you could imagine that this covariance matrix is sort of structured, and there are different molecules in both of those. You could imagine that this covariance matrix has a block here, a block there, and a block there. And that you could also imagine putting some correlations. Maybe jackfruit stands always next to the gas station, you know? I don't know. So you could put in a little bit of correlation. So actually, this is not as bad as it sounds. It's not great, but it's not as bad as it sounds. What's that?
Just a stupid joke. I said, should we say the gas station?
Gas station.
The Gauss station. Oh.
Yeah, that's why I said very.
May I offer to you, Kawe, that you may have seemed really cool, but you really are a nerd like me. Right. So the next thing we'll do is let's linearize the competitive binding model. And that's partly because there's so much stuff we don't know anyway that I'm not going to fuss about a little bit of details of this and that. So we're going to, do you remember how there's a threshold and then there's a saturation? I'm going to take the part in between and just draw a line through it and, you know, kind of get a plane and then look at that. So then I'm going to get, and we wrote down this model already earlier, that we're going to get that RA, the response, is KA. Remember, we're taking the summed response of the receptors times a sum on I of SAI. SAI is the sensing matrix, so it tells you the response of the receptor to a unit concentration, and that you can read off by sort of you take the slope out of the competitive binding model in the middle, and it'll give that to you. So you get this times CI, that's a sum, and we agreed we're going to add Gaussian noise. That's probably kind of okay because there are many independent noise sources that sort of add. And then we're going to multiply this by square root of Ka because we're going to look at, I know there are many receptors and you sum the responses, you know, and there you go. So that's our model. And we're going to now calculate. And here's our calculation. We want to calculate, okay, so I'm going to parameterize this. So roughly speaking, we've said that the response vector, whoops, ouch, that the response vector is equal to a vector, a matrix that I'll define in a second. I'm going to call it K, and I'll tell you what that is in a second, times the sensing matrix times the concentration vector plus a noise vector. I'm going to write out the noise vector again. And the noise vector is equal to eta 1 square root of K1, eta 2 square root of K2. And I'm also going to ask you to recall from earlier in the lecture today that we gave different noises to each receptor because it's possible that basically the variance in the noise is different. But we're not going to allow the noise in receptors to co-vary because we're assuming that it's independent noise. Okay, I'm saying all those things because I will now want to write down K as being some diagonal matrix with K1, K2, Km, and this is the receptor populations. I'm defining these objects for a reason because the answer will get written in terms of them. And then I'm going to define sigma, which is a diagonal matrix of the noise variances. So I'll write it that way. And I'm going to understand, so each of this is eta A is a Gaussian with variance sigma A squared. My model is C1, C goes in here, out comes a channel, and out comes this. And this response model is used to compute that output, right? So basically there's a compression because S is a matrix that is M by N. It's a rectangular matrix. So there's a compression of the data and the orders as previously stated, and there's an added noise. So it has both of those phenomena. So now we need to compute the mutual information. I'm going to define one more object which is going to be useful to us. I'm going to define S times gamma. Remember, gamma is the covariance matrix of the environment. S gamma S transpose. And I'm going to call this the overlap matrix because it kind of quantifies the following thing. So you see S here is a sensing matrix. It's going to tell you which receptors respond if I give an odor. But if I have a given odor, the covariance matrix tells me if I have this odorant there, some other odorant is there too. So because of that, there's a certain degree of overlap that you'll get between responses in different cases. So this kind of quantifies the overlap in the responses that you're going to get. Okay? So I've named it that. Anyway, now we need to calculate the mutual information. So we have I of C given R, C and R. And I'm going to remind you what this was. It was the entropy of the odor distribution, which is just a Gaussian, right? Easy to do. The entropy of the response distribution is kind of also a Gaussian, right? Because this is a Gaussian multiplied by a simple matrix, so it sort of just re-sums the Gaussian in some slightly annoying way. And then you add more Gaussians, right? So this is kind of a sum of Gaussians, so this can be handled, and you can do that integral. And then you have the joint distribution H of C comma R, which also, you can, this is not, as they say, not rocket science, but it takes a certain amount of willingness to sit down and write out, half a page very carefully of notes. So you can do those sums. They're all integrals here because C and R are all continuous. And you get a beautiful looking answer. So the answer is 1/2 the trace in the matrix sense of the logarithm of the following matrix. I here is the identity matrix plus K times sigma inverse. Sigma is the covariance matrix there, times Q, which is the overlap matrix. And that's our answer. It is a closed form answer because everything was Gaussian. Okay? Cool. So our goal is to see, we're going to end this lecture. Oh, I'm at time, but if you give me 5 minutes, I can tell you what the outcomes from this are. So this actually predicts a whole bunch of really interesting qualitative phenomena that are kind of in sync with different kinds of experiments and data, just by itself, with all the approximations we've made. So I just want to tell you that, what those things are, partly to show you that this kind of way of thinking carefully about what things matter in the system and then making a model which even cuts many corners can actually give you plenty of qualitative insights. So we want to ask, how should the repertoire be organized? That was our question. So let's go ahead and answer it now. So our goal is to compute these optima. We take di, d mutual, dr. So I maximize the mutual information with respect to the proportions of all the receptors. So Ka is bigger than 0 for all A, and the sum on Ka is equal to K. So with these constraints, I perform this maximization. So that's a standard maximization to perform. In general, you'll need, unless you pick the covariance matrices and everything very carefully, you'll need to do this numerically, but okay, do whichever one you please. And I'm going to tell you what the results are. So the results, and it's just a series of pictures, are as follows. So the first picture is this. So it turns out that this is, I'm going to draw the correlation between receptor abundance in this theory, abundance, and diagonal elements of Q inverse. So Q inverse is a matrix whose dimensions match the number of receptors you have. So if you draw this correlation, it turns out to be pretty close to 1. So the reason I'm drawing it like that is the inverse matrix is a very complicated thing, right? An inverse of a number is 1 over the number, but if you invert a matrix, then an entry in the inverse matrix is kind of a function of the whole thing, right? It can depend on everything. So what we just learned from this is that the abundance of receptor A depends globally on all the receptors. So the reason why I'm emphasizing this is a very common way to think in biology and elsewhere is suppose you have a bunch of receptors and they're responding to a bunch of odors. And let's suppose receptors 1, 2, and 3 are responding a lot. Then you would think, oh, you should have lots of receptors 1, 2, and 3. You can lose the others, or you have less. That is just totally not true, because this depends on the inverse of this matrix. It's not the sensing matrix itself, but it involves the inverse. And the result is it depends globally on the whole thing. That's because it may be that 10 weak responses tell you a great deal more than one strong response. So you may actually not care about the strong response. You want those 10 weak responses, right? So that can happen. So this is important because there are a whole bunch of experiments that were done which were sort of uninterpretable, because things would happen and then everything would change in the receptor proportions and things like that. And what's going on? Well, I think this is the reason why, that the actual answer for how you want to design the system depends globally on the whole thing. So this is not a simple increase the proportion of highly active receptors. Receptors rule. Okay, that's result 1. So that's a statement about how we should be thinking about the olfactory system, that we shouldn't be thinking in terms of which receptors respond a lot, but somehow holistically in terms of the entire code at the same time. So that's important to keep in mind. A second result is that diversity increases with the number of ORNs. So you can draw a plot here. So the way you do this is you pick some sensing matrix, either taken from data from fly or mouse, so you just make it up, use some random sensing matrix. And you'll find in general the number of ORNs and the number of types that you should include. So it goes like this, in a slightly sporadic way. So there isn't very much actual data, but there's some. Here's marmoset, here's cat, sort of mouse, there's a guinea pig floating around here. Anyway, there's data for a few, okay? And on the x-axis, if you say how many olfactory receptor neurons do you have, I mean, that's not really available. So what you do is you take the area of the olfactory epithelium as a proxy, and you might say, great, that should be fine, but actually there's an epithelium. But actually, there are various studies that show something called allometric scaling. If you double the size of the brain, you don't get twice the number of neurons. It sort of gets a division by mass to the power 0.2. So no one has measured the number of olfactory receptor neurons in all these species, unfortunately. So I'm going to take this as a proxy. I mean, we're just sort of getting some sense of whether this kind of makes sense. And on this side, there's some notion of the number of types. For some of these species of small number, somebody has measured the different numbers of types, and you get a kind of increasing line. I don't mean that as being any definitive match, and I'm not even going to try to match the slopes. There's too little data here, and this is with some made-up receptor repertoire. In fact, every animal has its own receptor repertoire, so really we should do this for every animal separately. So we haven't done that, right? So I just mean to say that there is a rough qualitative match. The third thing is the most important one. So you can take an environment 1 and environment 2. And here I can put in all the, this is the covariance between the odors, sorry. This is the covariance between odor 1. So here I have all the odors in the environment, and here I have all the odors in the environment. So this is the covariance, or this is the odor covariance. You remember we have that for the environment. So I'm going to draw that, and the diagonal elements are here, the variances. Okay? So imagine I make one environment where, let's say, this one and this one and this one have a large variance. And then I make an environment 2 where this one and this one have a large variance. And the other things are sort of floating around. Easy to do in the lab. You just change the variance of the odors you present. This is what you predict will happen. And by the way, it's amazing that we can even check such a thing, because normally in the eye, for example, the proportions of photoreceptors are wired at birth. But I told you that the olfactory receptor neurons die every month. And just like 6 years ago, there were a series of findings reported that, for some reason, when I look at animals that have been exposed to different environments, I seem to be finding that the proportions of different receptors are changing. And people are like, what? That makes no sense, et cetera, et cetera, et cetera. Okay, so this theory is, of course, going to predict that if all your receptors die and you grow them back again, that it would benefit you if they to change their proportions. That doesn't explain the mechanism, but it says that that would benefit. So what does it predict? And this prediction is qualitative because, we have to go species by species by species. And in fact, because of this, this fact that we said, in order to make a precise prediction, we need the entire sensing matrix. It's no good to tell me that you give me 50 out of 500 receptors, because it depends globally on all the receptors. So I have to make a qualitative prediction because we don't have the precise data. What you find, that if you look at the abundant receptors, so this is the abundance, and this is environment 1, and this is environment 2. If you look at the abundant receptors, they kind of change a bit, like that. But if you look at the less abundant receptors, you predict massive changes, like that. There are very big changes in proportions that are predicted. And there's another interesting thing that you predict, that if the experience changes a little bit, so that no two individuals are going to see exactly the same variances and stuff, because they would explore the environment in an experiment. The animals explore the environments a little bit differently. So one way they do these kinds of experiments is they'll put the other odor in the water. Well, some animals drink more often, so the actual experience varies a bit. So it will turn out that the actual change, the error bar on exactly how big this change is pretty big. So if you change a little bit, well, a reasonable amount, what the experience, the variance is, you can change this quite a bit. There's large variances here. So let me put it like that. So now I'm going to describe data from the experiment of Ibarra, Soria, et al., which came out in 2016, where they did the following thing. They raised a bunch of mice, and they would expose the mice, the same wild-type mouse. They would expose the mouse to different olfactory environments by putting the odor in the food or the water or something, and they would change what odors they were going to put in. They would allow the thing to go through a whole cycle of losing its epithelium and getting the new receptors. Then they would sack the mice and RNA-seq, grind up its olfactory epithelium, and RNA-seq the whole thing to figure out how much expression you have of the different receptor types. That was the experiment. And this is what they found. So this is the log fold change between environment 1 and 2. And this is the RNA-seq counts, which we're going to take as a proxy. We're going to take RNA-seq counts as a proxy for how many of the receptors you have. And you see this. So this is for about 1,000 receptors. And then if you look at the error bars, or the variances between individuals, they're large here and get smaller and smaller here. It's like that. So I would claim to you that that's exactly this qualitative pattern. So we could discuss things like mechanisms for producing these changes and what it takes to make a detailed prediction. We just need a lot more data. But I think this is a very nice, at least qualitative theoretical explanation for something that's otherwise a complete head scratcher. Why would these mice have such different receptor repertoires? That's the end of my lectures. There's a lots more to say, but this is supposed to give you a flavor. I want to remind you of, we did CRISPR and we did the olfactory system, both using basically thinking about molecular sensing. We saw many different techniques. We saw, dynamical systems, population dynamics type techniques. We saw various kinds of statistical techniques for modeling things. We saw information theory as a technique for describing systems, classical molecular biophysics as a way of doing things. And really, when you do this kind of theory, you should be willing to take any technique that will help you. If it helps you to sort of paint a picture of it, that's what you should do. Right? And you grab all the techniques and use them to solve the problems. A pleasure teaching you. See you soon. Lunch.